My name is Katie Greenham. I am an assistant professor in the Department of Plant and Microbial Biology at the University of Minnesota. I'd like to first thank the organizers of this seminar series for inviting me to give this presentation, and thank you for taking time out of your day to watch this recording. Today I'm going to go over some of the research objectives of my lab and some of the approaches we are using to achieve them, and convince you, I hope, that it's really important to think about time of day and the importance of daily changes that happen throughout the day when thinking about your experimental design. We are really interested in the circadian clock, this internal oscillator that's present in all organisms that synchronizes internal processes with the external environment. Examples in plants can be flowering time. This can be daily and seasonal regulation, metabolism, responses to abiotic and biotic stresses, and hormone signaling, and ultimately coordinating all these different processes to maximize growth throughout the lifespan of the plant. And we really are interested in how these signals are perceived by the plant and then how they're responded to in a time of day dependent manner. Responding to abiotic and biotic stress is very energy taxing, and the plant cannot mount a defense response 24 hours a day and still maintain growth. And how that response is then mediated at different times of day can have implications on ultimate fitness under stressful conditions. Much of these responses occur at the level of the transcriptome, and a recent study by Ferrari et al. Really highlighted the importance of this daily and diel cycling of the transcriptome. And this was a study done across the plant kingdom and really looking at the conservation of circadian and diel regulation of the transcriptome. And it really is universal. So a lot of the genes that are involved in photosynthesis, for example, naturally will be expressed during the day, but really it extends beyond that to all levels of the transcriptome. And you can see in these heat maps below these different, across the green lineage, you can see that genes are expressed in alternate times of day, where you have genes that are expressed predominantly during the day and then genes that are expressed at night. So really understanding this full time of day response of the transcriptome is really important to understanding the full breadth of response. Another reason to be interested in the clock is there is mounting evidence to support its importance in crop adaptation, not only just for crops, but also natural populations of plants. So there have been several studies done on crops. Examples shown here are barley, tomato, soybean, showing that as crops were domesticated and favorable traits selected for, the genes that underlie these traits, many of them are involved in the circadian clock. And so this kind of highlights the importance of thinking about the clock when selecting for traits of interest, and that the clock is actually a really good target for crop improvement because it's already integrated into the physiology of the plant. And it makes sense to, when optimizing certain traits, to target the clock itself, which will hopefully enable plants to maintain their fitness while also adjusting certain responses of interest. So the study here shown on the bottom in 2015 showed that for many of these different crop varieties, a lot of the traits that had been selected for as they had been expanded in the latitudinal ranges during domestication, a lot of those genes that underlie those traits are circadian clock. And many of these have been predominantly for photoperiod selection. As you expand to different latitudes, it's really important to adjust flowering time in order to complete the life cycle within a different growing season. So because we know the clock is critical for flowering time control, it makes sense that a lot of these genes are involved in the clock. But I think it extends beyond this now as we think about other stresses and we go back into natural varieties of some of these species, that we think about the clock control of those processes as well as we try to breed them into the current cultivars. And this really is the basis for what we do in the lab. Is we're really interested in some of these main questions that I've listed here. These are kind of the driving questions in the lab. One is to understand how daily processes are coordinated in the plant to maximize fitness, and then how stress responses are integrated into those processes at the tissue and the cell level. Once we understand that, can we identify certain transcriptional regulators to alter the network architecture to get a phenotype that we want, whether that's enhanced stress response or any specific trait that you're interested in. Once we've identified those, we want to try to develop some gene-edited plants that temporally optimize stress improvement traits. And what I mean by that, I will hopefully become more clear later on in the talk, but really it's making adjustments to those regulators in a time of day fashion. Because everything we think about is over the course of a day, we think a lot about patterns and identifying patterns of interest. And this can be at the level of transcription, physiology, or metabolism, or really any trait that you study. For me, it's really thinking about the pattern of the response. And this means taking measurements throughout the day to capture these dynamics in either gene expression, in this case, stomatoconductance, which is one of the physiology measures that we use, or metabolite levels. And typically when I show any of these graphs of data, you're going to see these bars on the bottom. And these will indicate the day and the night. So the white bar will always indicate the day. The black bar will indicate the night. If an experiment is done under a circadian condition where we remove the light dark entrainment cues, it will be under constant light. And this gray bar will represent the subjective night. So this is the night that the plant experienced before we shifted it into constant light and temperature. And this condition will capture any cycling or rhythmic patterns that are driven by the clock itself. And so that we've removed any temperature or light signals that drive those oscillations. Once we have these patterns of interest, we then want to associate those patterns with a certain trait. And stress is going to be the one that I talk about because it's what we focus on the most in the lab. So you can imagine having a series of patterns that a more tolerant crop has. So this is a more cold tolerant plant. We see these patterns. And then if we have a crop that's sensitive to that stress, it will have a different set of patterns. And then the idea is to identify key patterns at the level of transcription that we've associated with the physiology and metabolic outputs that we think we can integrate into a different crop to modify its physiology to be more tolerant like this other crop. So that's really the ultimate goal. So as I mentioned, I'm going to give you a few examples that will highlight the approaches that we are applying to tackle these questions. And these are the takeaways that I hope you will get and I will convey during this talk. And the first is really to convince you that patterns matter and that classifying gene expression based on patterns really uncovers some new insights into transcriptome dynamics. Using pattern analysis, we can also learn some really interesting things about polyploidy in crops and what retained paralogs in crops such as Brassica Rapa, which is one of the systems that we look at in the lab. What are the implications of this expanded genome in crops, and what are the roles of some of these retained copies when we compare to what we know in Arabidopsis? So using pattern can identify some new levels of regulation. And then taking these patterns and building gene regulatory networks, we can identify potential targets for new functional roles following the divergence from Arabidopsis. So this is going to be a story I'll tell you about comparing Arabidopsis to Brassica Rapa, and how gene regulatory networks can help us identify some of these transcription factor targets that might have some new important roles in stress response. And then finally, which we're kind of just getting into now in the lab, is looking at how some of these paralogs that might have new roles have interspecific variation within Brassica Rapa. So within a species, we see a lot of variation, and this is built off of these gene regulatory networks and things that we're hoping to follow up on. As I alluded to, Brassica Rapa is our crop of interest in the lab, and there's a number of reasons why this is a really attractive system for us to work in. The first is that it's in the Brassicaceae, so it's a close relative of Arabidopsis, making translational studies really feasible. You can take Brassica genes and complement mutants in Arabidopsis and vice versa, which means while the genetics can be a little slower in Brassica, we can still do a lot of work in Arabidopsis and learn a lot in parallel. The other thing that I really like about Brassica Rapa is its incredible morphological diversity. So within one species, you have Chinese cabbage and pak choi, leafy vegetable varieties, turnips, oilseed varieties, and other inflorescence varieties like rapini. So it's really a very, very diverse crop and offers not only diversity in terms of morphological phenotypes, but a lot of underlying physiological diversity in terms of stress responses. So we know that the Chinese cabbage and the turnips are much more cold tolerant than these oilseed varieties. So it's a really nice system to ask how stress response varies among these different morphotypes and whether we can use that to identify our targets that I mentioned earlier. To give you a little background on Brassica, it diverged from Arabidopsis around 20 million years ago, and since then the genome has gone through a genome triplication followed by extensive fractionation. As a result, in Brassica Rapa, you can have up to 3 copies of every gene in Arabidopsis, but again, there's been extensive fractionation, so in some cases, back down to single copy. So I mentioned the variety in Rapa. Oleracea is another brassica, which contains your broccoli, cabbage, kale, and Brussels sprouts. These Brassica Rapa and Oleracea are also really great models for canola, or Brassica napus, which is a much more economically valuable crop, especially in the US. So the canola here, also rutabaga is part of that, and Siberian kale, not shown here. So Brassica napus is an allopolyploid derived from hybridization between Oleracea and Rapa. So its genome is much more complex, which makes Rapa a much more appealing system to start out with to develop some of these models before transitioning into Rapa. But I just wanted to highlight that a lot of what we're doing in Rapa, we hope will also be able to be translated into napus. In terms of the circadian clock, as I mentioned, it's present in all organisms. And back in 2012, Ping Lu, a postdoc in Rob McClung's lab, where I did my postdoc, had just published a paper looking at how the circadian clock is expanded during plant evolution. So you can see here, shown in Ostriococcus, the green algae, to moss, Arabidopsis, and Brassica Rapa, a lot of the players in what we consider this core circadian clock have expanded. So here you're seeing in yellow, these are genes that are expressed during the day, and in gray, these are genes that are expressed during the night. So really early on in the algae, we have a very simple, much simpler clock. This has expanded a little bit now that we know a few more players involved, but in general, a morning and an evening expressed gene provide this feedback loop to generate these cycles or these rhythms. In moss, the CCA1 or LHY members expanded to 2 copies, and then we have an expansion of these night phase genes as well. And as you can see, you move into Arabidopsis, we have further expansion, and in Brassica Rapa, additional expansion with up to 2 to 3 gene members for each of these orthologs in Arabidopsis. And what Pink found is that in Brassica Rapa, there's been preferential retention for circadian clock genes in multiple copies, suggesting that the retention of these genes means that they are providing an important function. Many of these are transcription factors, so it's not necessarily surprising that they'd be retained in multiple copies. This could also have to do with a gene dosage effect, so maintaining proper balance. But it definitely suggests an importance of these genes in maintaining these oscillations, and also brings to light some new questions about what the roles of these new paralogs are in the clock, and if they've diverged in function or have gained new function that might explain some of the different traits or morphological variation that we see in crops, in particular in Brassica Rapa. When I started my postdoc, I really wanted to address that question and really understand what the circadian clock looked like at the transcriptome level in Brassica Rapa. To do this, I designed a very classic circadian clock experiment where I grew plants under entrainment conditions. So these are growing them under light, dark, or constant temperatures, that's LDHH for 2 weeks, or constant light and hot, cold temperature. And the goal of this is to identify genes that are driven by so entrained to a photoperiod or a thermocycle, and that are circadian regulated. So following these 2 weeks under these, what we call entrainment conditions, to synchronize them under these conditions, and then transfer them into free run, constant temperature and constant light, and collect tissue every 2 hours for 48 hours once they're in these constant conditions. So again, here's those white and black bars to signify entrainment into the free run. So the goal here is to identify genes that are rhythmic under these circadian conditions, driven by those photocycles or thermocycles entrainment. And so this will tell us what genes are controlled by the clock. To analyze this data, I took a co-expression network approach and used WGCNA, which is a weighted gene co-expression analysis, which I found to be a really effective means of isolating genes that have similar rhythmic expression patterns. So here I'm showing you the top 8 modules in the network. You can think of these as clusters of genes that have a similar principal component or eigengene that represents the pattern in that cluster. So these are the eigengenes, which again is like the principal component of this group of genes, to highlight what the predominant pattern is for these genes. And you can see for each of these modules, we see differences in the phasing. So here we have genes that are peaking in the middle of the day. Here we have genes that are peaking right at the start of the day. And you can see within these modules now we have separation for genes that have phasing or peaks of gene expression at certain times of day. What we found from this data is that 70% of the expressed genes were circadian regulated. So using the network analysis and also several other packages that have been developed to statistically identify rhythmically expressed genes from either microarray or RNA-seq data, we identified genes that have significant patterns of expression. So 70% of the genes were found to be circadian regulated, which is a really, really large number and was really quite surprising. So here I'm showing those 2 data sets. On the left is the photoperiod entrainment and the right is the thermocycle entrainment. And you can see here, these are the different modules. So the genes within those modules or clusters that have similar phasing and expression. And so I've arranged these based on their time of peak expression throughout the day, from the morning to the evening. And I hope you can kind of see these wave patterns as you move down these heat maps where genes have those specific phasings throughout the day. I then wondered whether, whether paralogs or genes that were maintained in multiple copies had any specific patterns. So do we tend to see genes that are in multiple copies expressed at certain times of day? And that might give us some insight into certain pathways that are enriched for retaining those copies. So here, what we did, because of the similarities between those two data sets, we actually combined the photoperiod and thermocycle data sets. And this is just showing you the eigengenes for the combined network, where again, we see that nice pattern of peak expression throughout the day for each of those modules. So then we asked for each of these modules, is there significant enrichment, over-enrichment or under-enrichment for having multiple copy genes or multi-copy paralogs within those modules? And what we found was there seems to be significant enrichment for genes that are expressed during the day to be enriched for multi-copy genes, whereas genes that are expressed in the evening, so shown on here on the bottom here, are under-enriched. And this is independent of the number of genes within the clusters, because in actual fact, some of these evening expressed modules had more numbers of genes than the ones during the day. So this is something that we're really interested in. We, of course, we've looked at the gene ontology enrichment, and a lot of these processes enriched during the day, light signaling, photosystem, photosynthetic response, abiotic stress response. But I think the caveat here is that a lot of the genes that are expressed during the night are naturally just not well studied. People tend to do their experiments during the day. So it's quite possible that a lot of these just don't have as much Go ontology associations as the ones during the day. So we take that with a little bit of a grain of salt, but it's definitely suggesting that a lot of these processes that are happening during the day are contributing to that retention of multi-copy genes. So it's something that we're definitely interested in following up on. So I hope I've at least convinced you that looking at these transcriptome changes at the level of the pattern or dynamics throughout the day is a really nice way of uncovering some interesting patterns and new insights into the important regulation that's happening, especially when it comes to the predominant regulation of the clock at the level of the transcriptome. Going back to those paralogs, and some of the questions, the next question was whether these retained paralogs are exhibiting the same expression pattern, or do we see divergence within these retained paralogs? So you can imagine a situation where you might have a set of paralogs that have diverged in phasing. So we see now one gene might be coming on earlier than its copy, or we might see similar phasing but changes in the amplitude, or it could be a combination of both. So we really wanted a way to pull out these changes. So in collaboration with Ryan Sartor, who's an NSF postdoctoral fellow at NC State, we came up with a new method to analyze these gene expression patterns. This is now available as an R package called Dipon for differential pattern analysis via linear models. And what this does is it builds on that co-expression network analysis that I mentioned that separates out the genes based on their expression patterns. And what we do is take advantage of this eigengene property. So this is that principal component that's representative of that cluster, and assign a value for every gene based on its relationship or correlation to that eigengene. And by doing that, we end up getting a single value that's representative of the pattern. So for each gene, it has a correlation score of how well associated it is with that eigengene for each of those modules. And those values then get put into a linear model, which allows us to test any number of treatments, genotype combinations. So any standard tests you would do with a linear model, we can now do that where our value, instead of a gene expression value, is a pattern. Once we do this, we can then run our significant tests and then cluster those outputs using the expression data to then see what predominant patterns are appearing for those genes that are found significantly different from the linear model. So when we ran this on our paralogs, we basically asked of all the expressed paralogs in the circadian data set, which came out to a little over 4,400, what percentage of them have diverged in pattern. And pretty surprisingly, it was over 42% of those paralogs have diverged. So this is kind of just showing you this a full view of the data here, where we have, again, our modules with our 2 copy genes. So this is 1 copy paralog versus the other copy paralog, and I've stacked them so you can really see the differences between them. And I just wanted to give you this full shot so you can kind of see the different waves of patterns. If I zoom in to 2 of them here, this is a little more obvious. So in this one module where we have, so this is, we've just randomly called one paralog BR1 and its copy BR2. So these are all the genes. So the top row here would be a paralog of the top row here, but they've been clustered based on similar expression. So this set of genes down here is peaking at the start of the day, whereas their copy is peaking middle of the day, middle of the night. Here's another one that's a little not as anti-phase, but a little more, a few hours earlier in phase between its copy. So not only are they diverging in phase, where in some cases we see them anti-phase to each other, but they're diverging together in sets of genes. So this really suggests an underlying regulation at the network level. And just to give you another example, so if we plot that data a little differently and just show the normalized expression for all those genes in those 2 modules I just showed you. So here's the expression of those genes we've called BR1, and then their paralog, their pairs BR2. So you can see in some cases we see that their expression is completely anti-phase, whereas in other cases we just see a shift in phasing. Definitely suggesting that at the level of regulation, the transcriptome level, we see pretty dramatic differences in the regulation of these paralogs. So this pattern analysis really uncovered some genome-wide phase shifts among these retained paralogs. And this is something that we are extremely interested in. And of course, one of the questions then is whether this divergence in phase, has one diverged from its ancestor Arabidopsis-like function, or are they both showing some new expression altogether? So we wanted to take advantage of the circadian data sets available in Arabidopsis and see if we could answer this question, to see if one of the pairs was more likely to maintain the Arabidopsis-like function based on its expression pattern. To do this, we used Genie3, which is it's a package in R for building gene regulatory networks. What this does is you provide all the transcription factors with their expression data, and then all the genes and their expression data throughout the time course. And what Genie3 will do is predict the target genes based on its expression. So one thing to keep in mind is this is not, this is independent of direction of regulation. So it is assuming that the transcription factor can either activate or repress expression. So when you see target genes, you can expect, this is a very simplified version, but you can expect to see target genes that have similar expression patterns to the transcription factor or anti-phase patterns if it's acting as a repressor. So the question here is, based on these expression patterns, can we identify which of these transcription factors in BRAPA is more like the Arabidopsis one based on its expression? Using the Brassica data that I've already described to you, we identified, for the genes that were expressed, a little over 2,000 transcription factors and a set of target genes. All of these had known orthologs in Arabidopsis that we could identify with confidence. And then using available microarray data sets provided by several different labs that have been curated on this diurnal database housed by the Mockler lab. And these are, we selected time courses that were similar to the conditions in our experiment. And from that, got a list of a little over 600 transcription factors and 9,000 target genes. So 256 Arabidopsis transcription factors with 2 corresponding paralogs were in common between these sets. We then built several different gene regulatory networks so the Brassica Rapa network, where again, we have our transcription factors that are in multiple copies, and they're being assigned a set of target genes based on their expression. And then a Arabidopsis network, where the transcription factor is being assigned a set of target genes based on its expression. Then using these two networks, we ask whether transcription factor A or transcription factor B in Brassica is more similar to the Arabidopsis based on the target genes. So just comparing the target genes between the two, which is more similar. And from this, we actually found that 49 transcription factors in Brassica had significant enrichment for the target gene set with the Arabidopsis. I'm not going to go into the details of the statistics. I'm happy to talk about this next week during the discussion. Essentially, we used a series of permutation tests to identify a significant score for the overlap in the target gene set. And so what was really reassuring is that among these 49 transcription factors that we identified, several of them are known circadian clock genes that we'd previously looked at kind of at the gene level to know that their patterns had been diverging. And we validated this with a number of other assays independent of that RNA-seq data set. And so what this really tells us is, this is definitely evidence to suggest that the divergence, at least for these 49 transcription factors, one of them is kind of maintaining its target set and regulation that we see in Arabidopsis, and the other one might have diverged and is controlling a different set of genes. Now, if this is the case, we wondered whether we could identify regulatory elements that might be driving some of these changes. And to do this, we looked back at a previous study that identified a large set of conserved non-coding sequences across the crucifer family and across the crucifers. And what these are a series of non-coding nucleotides that vary in length and that are found throughout the gene body, as well as upstream, UTR, downstream, and intronic sequences. So this is just showing you the distribution of these CNSs and where they can be found. So the largest, most of these CNSs can be found in the UTR regions, although there's quite a few that are in these flanking the exons. So these are the genomes that were used to identify these non-coding sequences. So what we did was take those CNSs and we wondered whether, instead of using the expression, we could actually use the CNSs to run that same analysis that we've done. So as an example, if we have a Arabidopsis clock gene, TOC1, that has two identified CNSs shown here in the promoter region, and then we have two copies of TOC1 in Brassica Rapa, one of them might retain both of those CNSs where the other one has lost one. So this is an actual example where we do see this case for these genes. The question is whether the loss of this CNS might be driving a different set of target genes, or whether losing the CNS means it's no longer regulated by a certain transcription factor. This is where Stephen Zorrick, an extremely talented computer science undergrad in the lab, comes in. And Stephen took that list of CNSs and identified the full set in the Arabidopsis genome, and then the corresponding locations of all those CNSs in the Brassica Rapa genome. Since we know that genes can be up to 3 copies in Brassica, he allowed every CNS to be present in 3 copies. And he identified just under 35,000 CNSs that are shown here in their distributions. Okay, so taking those that same network approach. This time, instead of the target gene expression patterns, we replaced the expression patterns with the CNSs. And we asked the same question. So based on, sorry, these should say the CNSs, but based on those target CNSs and those target genes, is the transcription factor in Brassica more like Arabidopsis based on the CNSs of the genes it's regulating? So this time when we did this, we identified 68 transcription factors. So more than even by the gene expression, 68 of the transcription factors of these pairs were more like the Arabidopsis ortholog, just based on CNSs. And again, we saw the same set of target genes that are the same set of, transcription factors that are known to be involved in the circadian clock. And when we compared it to the gene expression networks, we found 35 of those transcription factors overlapped, suggesting that these CNSs are predictive of gene expression and this architecture of the network. So this is really exciting to us because it really suggests that these CNSs might be really good targets for identifying what's driving these expression patterns that we're seeing, and gives us kind of somewhere to look in the future as I get near the end of my talk, and I'll explain why this is going to be even more relevant, but why it's going to be really important to identify some of these elements that are driving patterns of interest and how we can use that in some of our gene editing techniques. Now, getting back to the kind of the biology here and thinking about if we have two paralogs that are diverging in expression, and one seems to retain the Arabidopsis-like pattern of expression, but also regulation when thinking about the transcription factors. But what does this mean in terms of a physiological response, or is this biologically relevant? And we wondered whether this change in pattern might also exhibit differential responses to abiotic stress. So is it possible that one of the paralogs is responding to stress where the other one isn't and might be maintaining other functions? So does this allow for specialization of function among these paralogs? So I'm going to briefly introduce you to two data sets that we had available to us to answer this question. This is a drought data set where we grew this R500 or oilseed variety of Brassica Rapa. So I didn't mention before, but this is the same variety that we used for the circadian time course experiment that I've just been talking about. This was done under a light dark cycle, so diel conditions. And these plants were, so water was withheld 2 days prior to the start of this experiment. So this is at the onset of drought response. And what we did is took leaf tissue every 4 hours for 2 days for RNA-seq. And we also did a number of physiology measurements, so stomatal conductance and FV over FM, to capture the dynamics and physiological response. So you can see, similar to the expression pattern where we see these nice rhythmic expression patterns, we also see very specific time of day responses to physiology. In this case, for stomatal conductance, we found the most difference was at night. So in the droughted plants, we actually saw a reduction in their conductance during the night period. We did see some differences during the day. The FV over FM data, which I'm not showing here, we also saw certain time points during the day where we saw significant differences. But I show this simply to emphasize again that it's really important to consider the changes that are happening throughout the day, especially when trying to associate transcriptional changes with the physiology. Not only that, but depending on when you take your measurement, you may or may not capture a difference in response. The second data set is a cold response data set. So this was done on a set of 7 morphotypes in Brassica. This was again, under light dark conditions where we grew plants under normal conditions, and then, which was a 24-day, 14-degree night. And then we shifted half the plants into a cold 4-degree night, 10-degree day for several days, and collected FEFM measurements during the day, every 4 hours. And then for about one night, a full day, and one time into the following night, we collected tissue for RNA sequencing and metabolite sampling. So this was supported by a JGI Community Science Program grant. And from this, now we have a collection of FEFM data throughout the day, again, where we see time of day changes in response depending on the morphotype. So some responding more than others. So here again, this is 3 days worth of measurements where we have FEFM, and these are 4 different morphotypes, many of which are showing an increase in FEFM during the day under cold. But in some cases, like this pak choi variety, we see very minimal increase. So this is something that we're exploring as well. In addition to the transcription, we also have glucosinolate data. And this is looking at glucosinolate for the night and the day under cold and warm conditions. So that first data set, for the drought data set, we were really interested to know of those patterns, whether we see any separation in their drought responses. So the first thing we did was take just the control conditions, these well-watered conditions, and asked, of these paralogs, do we see differences in patterns? So before I was showing you, those were circadian conditions. So we, first wanted to test whether this pattern change we also see under more physiologically relevant conditions, so light dark cycles. And we actually see more of a difference. So before in the circadian data, there was around 42% of genes, paralogs with different patterns. Now what we see are 70% of pairs showing significantly different patterns, and 77% that are showing significantly different median expression. And so the median expression can be in the form of a pattern as well, or just simply one copy is just higher expressed than the other overall. So that was really exciting to see that this holds under these conditions. Now, of these genes that are showing significantly different pattern or significantly different median expression, do we see any enrichment for one copy or both copies being drought responsive? As it turns out, we see, in the case of the patterns, we see significant enrichment for just one of those copies being drought responsive. And this really suggests that at least for a set of these paralogs, they might in fact be diverging in function. So one of them is showing a response to drought and the other one is not. Similarly with this cold data, again, this is supported by JGI, and this analysis was done and is ongoing by Angela Roccono, a graduate student in my lab. And again, for the control conditions, we see similar percentage. So this again is now a third data set where we're seeing this. So this is really telling us that these are real changes that we're observing. And not only that, but we're now looking at a different morphotype. So within this Chinese cabbage variety, as well as at yellow sarsen, we're also seeing around 70%. So this tells us that this is not specific to this one genotype, but it's also occurring at the whole species level. And again, what we saw is significant enrichment for one copy being cold responsive for these pairs that are showing different patterns. This is also, again, just further evidence so that we have two abiotic stress data sets, one drought responsive, one cold responsive, where we see that these paralogs that are showing different expression patterns, one of them tends to be responding to abiotic stress and not the other. We have not yet looked to see whether it's the same one. This is really very new data that we're just kind of getting into, but that's definitely something that we're really interested in. We're also now, we need to expand to these other morphotypes because of course, there are a lot of differences between the cabbage and yellow sarsen. So we're starting to see differences, and I'll give a few examples in the next few slides to kind of highlight that as we are still getting into the analysis. So just kind of to remind you again, it's a good example is that cold data set where, we have that yellow sarsen and the Chinese cabbage variety. The The Chinese cabbage we know is very cold tolerant compared to the yellow sarsen. And so I've already shown you that we're able to identify these different patterns at the different types of data sets. And now we really want to identify which specific patterns are associated with a trait of interest that we can try to bring into this other crop. So in the last few minutes, I'm going to give you a few examples just to kind of emphasize what I mean by this, and then kind of end on some of the future directions. So as we're going through the data sets, we are going to use the same gene regulatory network approach that I talked about with the circadian data set. And we want to use that now to compare across the different morphotypes in Brassica to identify transcription factors that are regulating sets of genes that we identify as either cold or drought responsive. But while we do that, it's always good to go in and validate some known cold responsive genes. So this is from the cold data set. I'm showing you 3 sets of genes in the yellow sarsen and the Chinese cabbage data set. GI or gigantea is a known circadian clock gene that's involved in cold signaling. Core 27 might be familiar to some of you. These are well-known cold responsive genes. This particular one has been shown to be inhibitors of night expressed circadian clock genes. And then finally, this is just a light harvesting complex, light harvesting chlorophyll AB binding protein gene that's part of the PS2 antenna. And I put this here because we have, FVFM data. And so it's kind of nice to relate that, excuse me, to that physiology data. So on the top here, blue is in cold and the red is our warm. And again, this is our night and daytime. So you can see here, we see really nice patterns of expression for many of these. And in the case of the yellow sarsen, we see kind of a shift, a phase shift in some of these. GI is coming on at a much higher expression at night, and the core is also coming on higher. Minimal effects on this light harvesting complex. Where this Chinese cabbage, we're seeing a little more of a shift in the GI, less of a response in the core, and actually more of an upregulation of this light harvesting complex here. So these are just examples of some of the expression patterns that we are starting to see that we'll eventually try to associate with measurements like this FVFM data, where in both cases here, we saw an increase in FVFM for the cold conditions for both Chinese cabbage and this yellow sarsen variety. Similarly, when we look at our glucosinolate data, so this is again data that was generated by the JGI. And so this is very new data that we're currently analyzing, but we have been able to identify several aliphatic glucosinolates with confidence. And what's really interesting is we're already seeing differences within and across species. So in the case of yellow sarsen, we're seeing an increase in these 2 glucosinolates. More so, this first one at night, we're seeing an increase compared to the warm condition, whereas the Chinese cabbage is only showing a change in one of these aliphatic glucosinolates, but it's still not nearly as much of a difference compared to the warm control as we saw in the yellow sarsen. And here I'm showing you the expression of some genes involved in glucosinolate synthesis and sulfur assimilation. So SLIM1 that has 2 copies in Brassica induces sulfur assimilation and represses glucosinolate synthesis. And then APK1 is a kinase that's important in the biosynthesis process. So this clear upregulation in this yellow sarsen variety of this kinase is really interesting to us. And we see a little bit of that in this Chinese cabbage, but not as dramatic. And again, here's another example of a gene that has 2 copies that is showing very different responses, both at the level of the control patterns of responses, but then also their stress response, where the cold is showing a huge upregulation for this one SLIM gene. Whereas in this Chinese cabbage, we see a little response of this one copy here that's similar to yellow sarsen, but this other copy is not showing much of a difference at all. Now, once we identify, those target genes that we're interested in, So here I'm just kind of giving you a schematic to show what the goal is and how we want to use these patterns. So once we identify the transcription factor pattern or response that we're interested in our Chinese cabbage, for example. So we might have a number of different Chinese cabbage genotypes that show a certain response compared to yellow sarsen in response to stress. When we compare those patterns in the two genotypes, so here's our yellow sarsen, we see that it has this earlier shift. And we want to basically take this Chinese cabbage response and put it into the R500 genotype. So what we'll do is I'll identify a stress responsive promoter that has a pattern that we're interested in, the pattern that we've identified in the Chinese cabbage. And then we will put this into our yellow sarsen variety. So this is the yellow sarsen normal expression pattern. By introducing this promoter upstream of that transcription factor in R500, we hope to shift then that expression of that gene, that transcription factor of interest, to mimic the Chinese cabbage pattern that we've identified here as being important for that stress response. So this is similar to kind of overexpressing a stress responsive gene that gives you a stress response, but often comes at a cost of yield, because as I mentioned at the very beginning, mounting a stress response is energy taxing, and doing it 24 hours a day will limit growth. And we're hoping that, we've identified, and I hope I've convinced you that we've identified these time of day changes in response to stress. Many of these changes come at the level of time of day shifts in response, whether that's the phase or the amplitude. And we think that we can identify these phases and make these adjustments to small temporal changes that will lead to stress responses without compromising fitness. So at a broader level, we think this applies to really any trait of interest, whether that's involved in stress response or any yield output at the crop level. And as we think about going back to these wild varieties, we have more and more data at the level of the pangenome now that has shown us that a lot of stress responsive genes have been lost during cultivation and domestication. And so now we want to go back and bring these genes back into the cultivated line. But I think it's really important to remember that many of these genes will be controlled by diel and circadian regulation. And as I mentioned earlier, during domestication, we selected for modifications to the clock to allow these plants to survive in different environments. And that means that our wild ancestor clock is probably operating a little differently than the cultivated crop. And so while we bring these abiotic stress or any trait of interest, any of these genes into these cultivated crops, I think it's really important to think about the state that the clock is in that wild ancestor as we bring those in. So I say a clock-minded approach. And that was basically summing up the last takeaway here was that I hope I've convinced you that among those retained paralogs, we're seeing altered responses to stress as well as interspecific variation within Brassica, suggesting that it's really important to think about within each genotype what the endogenous clock or diel regulation of the transcriptome is doing and how we can use that to alter the transcription to get a certain output that we're interested in. And with that, of course, I want to thank everyone in my lab. And this is kind of the state that we've been in for a while now. I talked about Angie's work, as well as Stephen Zorrick is a really talented undergrad in the lab. And then Maddie is a new postdoc who's joined us. And then I've just been so fortunate to have a really fantastic group to help get us through this past year. Kat Markham is another postdoc in the lab. Ananda Menon is our lab manager and research assistant, and then some really fantastic undergrads, Tommy Neary, Layla and Eileen, and then Erin Lee, who's a graduate student in another lab who's jumped in on our lab meetings from time to time. a number of really fantastic collaborators, Ryan Sartor, who did a lot of that transcriptome analysis with me. Chris Pierce for ongoing support with Brassica resources. Of course, Rob McClung, my postdoc advisor. And then I really cannot thank enough the wonderful people that I've met since I have started at the University of Minnesota. Adrian Hedgeman, who's been fantastic collaborator with metabolomics work and our glucosinolate stuff, which I touched a little bit on, but didn't have time to talk about. Clay Carter, who we've been working with on some other exciting projects in terms of glucosinolates and nectar in Brassica. Rui Kwong is in the computer science. We've started a new collaboration with to help with some of these transcriptome studies, especially as we start to look at single cell studies in plants. Corey Hirsch is in plant pathology. We've been working very closely to develop some phenotyping, some high throughput phenotyping assays to track stress responses in plants at a much more high throughput level. Nathan Springer, my neighbor here in bioscience, who has been just a really great mentor and hopefully much future collaborator as well as we start to think about these regulatory elements and stress responses. And then Fong, who's been really helpful in developing some gene editing techniques in Brassica. And thank you for your attention. I'm sorry if I went a little over on time, and I look forward to speaking with all of you next week.